large construction sites, as well as the sites of natural disasters. And what they do is they will drill very deep bores that could be much as 100 feet to 100 meters deep, and they will install in these bores inclinometers or piezometers that are measuring various properties of the soil and of the geotechnical subsurface structure. They want to see, is it shifting and how stable it is, and pressures are present. And what they typically will do is they'll drill one of these bores, they'll put sensors into the bore, they'll fill it back up with some bentonite or some mud or sand, and they'll run cables all the way up to the surface and then put a radio on a cap at the top and collect the data from those sensors via an RF link to radios. Unfortunately, the bores will leach, wires will fall, and any of the sensors will be lost. So geotechnical community became interested in subterranean wireless communication. You've seen maybe some papers that look at doing RF. Fortunately, RF oils can go, distances of 10 centimeters to maybe a meter. After that kind of distance, you very quickly fall off at more than 100 dB loss. Whereas acoustics, especially low-frequency acoustics, can propagate much further at the expense of lower data rates. Some other applications that my geotechnical colleagues are interested in have to do with these large centrifuges that they use for earthquake testing. They put sensors in there and they run wires and cables through. As you can imagine, in those around at very high velocities, a lot of things break. So they very much like to be able to do things wirelessly. And there are some of the centrifuges are sufficiently large that RF approaches will also have some challenges. So we decided to do a couple experiments. We got an eager grant, the National Science Foundation, to look at soil acoustic communication, really do a proof of concept. You see here on the left is a ice fall day in central Illinois. You can tell by the maximally flat landscape that truly is Urbana Champaign. In the center, there's a tank, a trough that we used and filled with sand and some water, some early testing indoors. We went out to the field site. The center, you'll see an aerial view of the plot of land that's by the University of Illinois, actually by the Electrical Engineering Department, a field station. And there's a number of locations where sensors were installed in bores that were drilled into the ground. Some of these were as deep as 6 or 7 feet deep. Some were just a few feet deep. But going off in two different directions, distances up to 50 meters. To the right, you'll see some sample frequency responses. The vertical scale is the amplitude of the received signal in dB when a broadband signal is transmitted, and the x-axis is frequency hertz. The top rightmost figure is a received channel response 15 meters from the transmitted source. The middle one is 25 meters from the source, and the one is 35 meters from the source. And for these particular examples, the source was a subwoofer, similar to you might have in your theater system. It was operating at a center frequency 10 hertz using QPSK, so 2 bits per symbol, quadrature phase shifting. It had a 30 hertz center frequency and 10 hertz of bandwidth. 10 symbols per second centered at 30 hertz. Received signals over the distances shown here were decoupled at bit error rates 1 in 10,000, and we were able to get about 20 bits per second at 35 meters. And in order to get reliable communication at 50 meters of distance, we needed to drop down to 10 bits per second. Here are some of the receive installations on the far left. At top left, that the circle that you see is the signal as it's received after it's been base baited, so brought down from 35 hertz down to DC and sampled at rate. That's the quadrature and in phase components of the received signal. at 15 meters. The center top see the decode and equalize. So we've been able to, with digital signal processing, unwrap received signal into the transmitted signal installation. So if it's the upper left, for example, that would correspond to a 1, upper right would be a 1, 0, lower right would be 0, 1, and lower left would be 0, 0, for example. So just depending which of those constellation points we send, sending 2 bits of information. And in the upper right, you see the pulse response estimated from the data and training that was sent at a distance of 15 meters with that source. The center figures correspond to the same kind of signal at 25 meter distance. The lower 3 figures correspond to the same figure, or the same set of figures at 30 meters. So you see that we get a much more dispersion as we get out 35 meters. A lot has to do with the frequency-dependent absorption, getting a lot of attenuation at the frequencies out to 35 meters. We're all getting more multi-pack. There's more opportunity for interfacing with both packed clay layers as well as surface modes that are interacting with the surface layer. A figure that sort of shows a number of experiments, data from a number of experiments that we've undertaken over the last 6 months to a year. In our left, you'll see a speaker that is the UW30. That is an underwater pool speaker, so you can buy that on Amazon for about $50. It's watertight, so we buried that and used that to get distances on the order of a few meters to 10 meters and data rates upwards of a bit per second. In the center, we had enough experiments where we upgraded the underwater speaker to a higher quality pool speaker. That's the A339. So those are able to get out to distances of the order of tens of meters, data rates on the order of hundreds of bits per second. We wanted to get much further distances. We used a subwoofer that's a butt kicker. Is meant to be attached to the legs of sofa for use in a home theater. It is not to be waterproof, so if you see to the far right, we've encased it in lucite and sealed it with some putty, and then the wires out, and then buried it and used it as a vector. And we were able to get data rates at 2 bits per hertz, but get distances to 50 meters. We had some challenges with overheating, but it was certainly something that was manageable for this kind of experiment. Was repeatable. And the power to use to transmit for these experiments was substantial, much more than we would want the ultimate use cases. So we've been experimenting now with some small form factor, low power piston type transducers, and don't have experimental results to show you with those, but just want to give you an idea of what a Champaign looks like for a subterranean communication experiment. There's some drilling and some digging. They're playing with drones and taking video. There's testing out in the field. I even managed a shot here with Buck out in the distance, who was visiting on sabbatical and helping out with some of the data measurements. This was a nice crisp fall day. We collected some of the butt kicker experiments. And one of the beauties of living in Central Illinois is we get beautiful sunrises and we also get beautiful sunsets. So with that, I finish my part of this 2-tier talk and hand it over to the next speaker.
All right, thank you, Andrew. Maybe what I'll ask for those that are in the virtual audience, if you have questions, there is a chat window that once we get to the end of Shira's talk, that you could potentially include your questions and we can get the speakers to answer those as they come up after Shira's talk. So Shira, do you want to unmute your mic and turn on, there you go, perfect. Yep, sounds good. So I see somebody came in with a chat question. We'll hold off and address that at the very end. Andrew, would you be willing to hang out and answer some questions at the end of Shira's talk as well?
Sure.
Great, great. Thank you again, Andrew. So
Okay.
Shira Gordon, thank you for joining us today from sunny, I'm sure, and warm Parlera, California.
It is indeed sunny and warm here. All right, so you guys can see my screen okay and hear everything all right?
Yes, looks great.
Okay, so this is actually what the view, not today, it's a little more cloudy and dusty, we should say, in the valley. But this is Central Valley, California, where we do a lot of agricultural work. So before I get started, I just want to acknowledge the rest of the team that worked on this. In the center with the hat is Rodrigo, and so he is one of the main scientists here at the USDA. And then the other two are, so next to him is Valerio, and below that is Raquel, and they are two of our Italian collaborators that have done a lot of the work on this. So with that said, I'm going to tell you a little bit about glassy wing sharpshooter, GWSS, and Xylella as the problem, their communication system, and then how we're actually using vibrations as a control mechanism. And then I'll end with a little bit of new work that we're doing on signal transmission. So first, this is the actual bug. It's about 1 centimeter big, and it is a xylem feeder. It is native to the Southeast US, and importantly, it transmits this bacteria that I mentioned called Xylella fastidiosa. So as this bug lives, it actually lives on a variety of plants, and it communicates through vibrations. So first, briefly about Xylella. Xylella is a bacteria that can kill a grapevine in 2 to 3 years. Grapes are a very economic importance here in California. These are pictures of what would happen with a plant that is affected by Pierce's disease, which is what the effects of this Xylella is. You can see here this center cell is actually filled with bacteria, and that is how the bacteria, so the insect feeds into the plant and transits this bacteria through its mouth parts, and they basically clog the vacuoles that the plant needs to spread water. So that's why in the previous slide the leaves look like they're brown and dying. Currently, there are several mechanisms that can be used to control the insect, one of which is pesticides. And usually that's an area-wide application. However, it has to be coordinated, because if one field has spraying happening one day but another doesn't, then the bugs can hop from one area to another. There also is, you can't spray during flowering time periods and pre-harvest intervals. So there are drawbacks when it comes to spraying the fields. Another mechanism that's used is using egg parasitoids. So these are all parasitoid wasps. And we're talking, these are 1 millimeter in size, and you can see each one is coming out of an egg in the upper right picture. Again, this method works, but it is only so effective, and especially in the winter when the insects are overwintering, you have a die-off of the parasitoids. So what we are proposing is, can we use vibrations? Can we understand the vibration system and use vibrations as a playback method to control them? We have developed a 3-step methodology, starting with description of their signaling, continuing with identifying a disruption signal, and finishing with an execution of how we can disrupt their mating. So to first describe to you a little bit of their mating, this right here is a female, and I'll play a video. Hopefully that volume is loud enough. I forgot to turn it up before starting. Can you hear that? Is it playing? So you see her abdomen vibrating? And that is basically how they signal back and forth. So she's signaling to an off-screen male. The male then will signal, and he has a little more dynamic. You can see here a big wing flip. And then he also does abdominal vibrations. And so they basically duet back and forth in this, and this is how they go through their communication system. So I have here on this next slide a spectrogram that shows the signaling. So on the right is a male, And what you can see, I'll play this one for you as well, but you can see here, this is where that big wing flip happens, and then we're looking at a scale of about 1 kilohertz, and the colors are the intensity of their signal at the different frequencies. So you can see here, is that loud enough for everyone? Hopefully you can hear it. The female signal, you can see hers is one that we've been using. And with her signal, you can see it starts low and goes a little higher in frequency. This is, again, about a 3 second long signal. And you can see her harmonics in the signal go up, and that's variable based on distance. So the first stage that we did in description was looking at what individuals do when they're alone on the substrate. And you can see here that the females are much more likely to call. They call 80% of the time when they're alone compared to the males, which are calling maybe 20, 25% of the time. We then put the male and female together as a pair, and this is what we consider phase 1 of courtship. And here, I won't play this one, but you can see we've got a male fixed signal, female signal, male fixed signal, female signal. They're going back and forth in this tight duet over, you know, a very long time period. In this screen, I've got two full minutes that they're just going back and forth like that. During this phase, they're usually stationary, and we consider it a species and sex identification phase. From here, usually the male starts moving to find the female, and this is when there is then localization and advertisement to show that they are good signalers and therefore good potential mates. And in this one, you can see the male signal, male signal, male signal, and then you have a big male signal, a female response, and then the male is walking. So it's a much more male and intense signaling time period. He is the one putting in more energy. Once they become close proximity, we now are in phase 2 of signaling. And this is when we consider it really the close courtship communication. And here you can see the male is doing a lot of these abdominal signals and then the big signal that you heard. And then finally the female gives a little signal here at the end. I'll see if I can play this one and if it'll show up, if you guys can hear it. It's starting right here. It's the male signal. Then he's doing these little beats with his body. That was a female, and then here's the male walking. And so from that, then they hopefully mate. And then what we found is on average, they mate for actually 15 hours. And we've actually found that they do courtship communication. And I can show you a video of this one. And you can see the male and female are connected. And during this connected time period, they go back and forth and they signal some. So that's interesting for communication purposes and why are they doing that? However, what I'm going to talk to you today about is their signaling. And this is focusing on the male-female pair as well as if we add an additional male to the signal system. So here's one parameter that we looked at. And this is looking at the duration of their signal. And you can see that female from phase 1, when they're both stationary, to female from phase 2, they're actually different durations significantly. The first part of the female phase calling is significantly longer. And I'll come back to that later in the talk, but that's just important to note. So looking at both duration and then a variety of frequency components, we did a functional analysis, and you can see in blue, the male signals all fall out together. The red, the female signals fall out together. And actually there's a male rivalry signal that falls out more similar to the female calls. So taking this information, we now know what their calls look like, and we can use that for an identification purposes of what will disrupt them. So we tested white noise, which on the left you see the female signal, on the right is a bunch of white noise that basically blocks all frequencies of the female call. We did female calls, and you can see here the P stands for playback. The walking is when she's walking, and then the F is actually a female calling with that playback. And then we did a female noise where you have basically female calls on top of each other, so similar to the white noise, but it's a little more focused in the frequency spectrum. And in this figure, you can see this was a trial done where afterwards, the male, as soon as the female noise was turned off, you can see a duet between the male and the female on the right. So we did these trials with females alone to understand how they were communicating, and then with mating calls, or mating pairs, sorry, excuse me. So first, to show you some results of female alone, here we have silence versus female calls, and 60% of the time after the first female call, within 3 seconds, there was a female response. And so what that is showing is that the females are actively replying and calling with our playback. So they're signals that are interesting to the females and instigating a call. With the white noise, what we did is we compared the females and their calling rate to when the white noise was turned off. And again, we found that as soon as the white noise was turned off, the females were significantly more likely to call. And this is a common phenomena in signaling, animal signaling across species, where when there's noise, perhaps it's wind in the environment, and then it's calmer, quieter, the animals will call. So that's a phenomena shared across animals. Next, we looked at mating behaviors, and you can see here, compared to silence, both the female and the white noise significantly reduced the mating. And this is looking at approximately 30 pairs per treatment compared to silence. With the female noise, we did it first where we had the two with the female noise and then turned it off. And when the female noise was playing, there was a significant reduction in mating. And then as soon as the noise was turned off, we had more pairs that then mated. So we know that the pairs were viable, and it was indeed our treatment that was reducing them from mating. So knowing all of this information, we went to the third stage, which is execution. And we decided to take all of these signals and use them sort of as a hybrid, where we did the female calls close together, as you can see in the bottom picture. We did this in the field at Fresno State. And what you can see here is we've got individual bags, and these are each on grapevines. And in this bag, so the bottom right, you can see there's two little vials. So we would put the bag and the vials in, seal it up, open up the containers, and let the bugs naturally live on the vine for a period of time. In the upper right, we have a shaker that was affixed to the metal post, and that would actually transmit the signal. So to show you how that worked is we had the shaker here, you can see in this vine right here, and then one vine away, which is, we would consider that vine 1, 2 vines away, that's vine 2, and so forth all the way to vine 6. This here is Rodrigo in the field. What we did is we actually did measurement of the signal, so similar to how in the agricultural field you might want to know how much pesticide is in the environment. This is to figure out how much signal is in the environment to affect the signaling. And then we have a portable computer and our station that we bring with us to do these measurements. What you can see here is at the shaker, the signal is very loud and intense. That's the bottom picture. And then the top at vine 6, about 36 feet away, you can see there's a huge drop off in intensity as well as frequency range. Both of those go up to approximately 1 kilohertz. At vine 3, you can see an intermediary level of intensity as well as frequency component. And what's interesting is we actually got a number of female calls, and you can see here the female call naturally next to the playback, and they're very similar. So we have a good representation in our playback of female signaling. So first, from the biological perspective, over 10 different field days, 134 mating pairs, we found that there is a significant effect of our treatment. We only had 1 pair mating in the treatment compared to approximately 20% or 28 pairs mating with the silence. And we did these on the same day, different rows. So 1 row had the treatment, 1 row did not. When we look at this from a sound transmission perspective, first we look at what we're looking at here is the FFT component. So we've got the frequency on the x-axis and the relative amplitude on the y-axis. You can see for vine 0, you have what we're calling a vanishing point, very far away, and it's actually well over the signal of the insect, but the way the signal transmits, it goes up to approximately 6,000 hertz. You can't see it in this picture on the right because it would be off the screen up way too high. If we look at vine 6, you can see that light green line is closer to 500-600 hertz where that vanishing frequency is. And so let me zoom in on that for you. And you can see that the light green is approximately 500-600 hertz, The female call in purple is closer to almost 1,000 hertz. And we actually found that the vanishing frequency of the natural female call is at about vine 3. So that's about where the female would naturally call at, and that's about where our signal transmission is. So that means vine 0 through 2, we're getting a signal that's louder, or sorry, the signal that has a higher frequency range than natural calls. And then after vine 3, so vine 4 and beyond, the signal has a lower frequency component. Next, we looked at the signal intensity based on the vine position. And not surprisingly, vine 0, they're highlighted in green, has the highest intensity, and vine 6 has the lowest intensity. And that's focusing on, each of those lines is 1, 2, and 3. So focusing on the main 3 frequencies, the harmonics that are transmitted. And in this, when we compare it to the natural female call, her call lies between 4 and 6, vines 4 and 6. And interestingly, the 100 hertz actually has a slower decay rate for intensity. So that means that its intensity is being transmitted further. I would expect further down the vines that 2 and 3 would separate out, where 200 hertz would be more intense than 300 hertz. But again, we're transmitting through the wire, so that's coupling it to those transmission properties. So I've actually talked about the first 3 parts of this talk already, and I'm going to go a little more in depth about signal transmission, which probably interests all of you. So just reviewing what I've talked about, we know higher frequencies do not travel as far, and that there is a drop of intensity with distance. This fits well with a lot of the theory out there. We know that under 5 kilohertz, the wave speed depends on frequency. So what we're looking at in this figure is here on the left, there's a big intense across all the frequencies input of signal. And then as time goes on, that signal dissipates. And then when we're 45 centimeters away, we actually have these higher frequencies arriving before the lower frequencies. So thinking about the wave speed and what our actual signal looks like, we wanted to know what's going on. So we have different harmonics, with the main frequency component being there in red at approximately 100 hertz. At some distance, we know that there's a lower intensity and fewer harmonics, higher frequencies being transmitted. But is there a point where that actually becomes such a drastic shift where at the beginning of the signal, there is no low intensities, the middle of the signal, all of the intensities are there, and then by the end of the signal, the frequencies again are missing the high component? And this leads us to the next question of what about that duration? So if you remember in the beginning of the talk, I said phase 1 female calls are much longer, and that's when the animals are further away and stationary. So perhaps our working hypothesis is that phase 1 calls are longer to accommodate for this timing shift during transmission. And when they get closer together, they don't need to have as long as the signal because they're already closer, and that transmission doesn't need to be as far. So we've done some preliminary work with this. Right here, you can see this is a shaker in the blue circle, and we've measured with a scanning laser the displacement across the whole grapevine. So they're gray right now, which means they're flat. As they go in and out of phase, they go from green to red. So I'm gonna go ahead and press play, and you can see the signal starts transmitting, and it's sort of had this long wave pattern, and it's moving in and out of phase across the branch. And the little branches coming off of it are gonna move not necessarily exactly as the main branch. So that's at 100 hertz. When we look at 300 and 600, and I'll play these together, you can see it's a shorter wavelength as well as slightly less intense, but it's a shorter wavelength and it's moving fastest at 600 hertz. That falls precisely with what we'd expect. That wave speed is higher for higher frequencies. So looking at this with real numbers rather than just a video, we can see here, this is now time. You can see here this first bit is where that signal actually is transmitted. And to zoom in on that, you can see that we've got signal transmission in blue and yellow are in and out of phase, and we've got this wave speed at this angle, which is basically the phase velocity of the bending wave on the plant. So we've got this wave speed, and when we compare the wave speed over 300 and 600 hertz, you can see that black line becomes more and more sharp, which is basically showing that we have an increase in this wave speed, which is expected, but very cool to see in our actual plant on the small scale transmission. So looking at that with averages over several plants, we can see this wave speed increase over increasing frequencies. So the phase velocity is increasing. So that's exciting in itself, but what's really cool is it actually fits with two different beam theories out there. And so that's exciting, and it's basically showing that the wave velocities level off at higher frequencies. So the differences between these two theories, you can see in blue and green. At the high frequencies, they have slightly different leveling off rates, but our data only stops at 1,000 hertz. So we can't decipher between which of those two theories. So with that, that's where I'm stopping the talk right now. I don't know who here might be going to the Acoustic Society of America meeting in May, but this last portion of the talk is what that is all going to be focused on. And should any of you ever come to the area in Fresno or Parlier, California, this is what our facility looks like if you ever wanted to come here for a visit. So thank you.
All right, thank you. Thank you, Shira. At this point, if any of you that are logged on, clap, clap. Yeah, I guess we should do the same thing here. If any of you are logged on and have questions for Andrew or Shira, this would be the time to type those in, and I'll read those. Andrew, are you back? Do you have your mic on?
I think so. Can you hear me?
Yep, I sure do. Also, UVM, how many folks do you have there? I'm going to make a note of how many we have in attendance.
I have Rodrigo here with me right now.
Okay. That's you, Shira, right?
Yes.
Okay. Let's see, UVM. So Kathleen is there. Thanks, Kathleen. All right. So if any of you have questions, just type them into the chat window, send them along. Sure, I do have one question for you. Instead of typing it out, I'll just state it since you're still logged on.
Sure.
So given the changes in the kind of the arrival time of the different harmonics that you were playing back, do you think that the animals are actually using information in the disparate arrival times as a cue to maybe distance to the sender receiver, or
Yeah, I think that there's a possibility that the signals that they receive that have very few high frequency components would indicate that they're far away. So if an animal receives, 100 through 1,000 hertz, then they have one sense of distance. But if they only receive up to 300 hertz, that's far fewer harmonics. But they still can, other studies that we have done show that they still are behaviorally responsive to just the low frequency components. If they're receiving well over 1,000 hertz, then they probably can sense that the animal that they're communicating with is closer. So I do think that that is a high possibility. As far as timing, that's exactly what we're looking into, and I'm not sure at this time.
Okay, great, thanks. We have a question for Andrew too, from UMass Dartmouth. Were the nonlinearity issues in meat comms in the meat medium or in the transducers? And is pork more or less linear than beef liver?
Good questions. So my collaborator, Mike Oles, believes that the nonlinearities are in the medium more so than the transduced, because he hasn't seen levels of nonlinearities in his measurements, but he hasn't sent a wide band of signals as we sent, especially for hero experiments. So it's clear it's either medium in water or in the transducer and its action, but the limiting factor in those very high bandwidth experiments was more so nonlinearity mitigation that was signal to noise. So it was signal to interference, where the interference, the nonlinearity, not linear interaction. If it was linear, our equalizer could take it out fairly easily. And something that we just haven't done at the time to get is, come up with better equalizer structures that account for, model that nonlinearity to try and take it out. So I don't know the answer, but I I can't say whether beef or pork were more linear. Certainly, the pork held together better water tank than the beef did. It never got pretty disgusting after a couple of hours.
I can imagine. All right. Do we have any other questions? There's a question for Shira from Louise Armith. How can the shaker vine system be scaled up to an entire grape field?
That is an excellent question, and that's exactly what we're working on. So right now we know that this goes at least 6-7 vines away, so 36 feet, and it's got no problem transmitting the signal at an intensity that is high enough to control the insects. We're working with industry to create shakers that maybe are more powerful, or perhaps just to learn the spacing that we need to use. And actually one component that we're looking at, we just installed in the past fall, so we're not ready to test yet, but we're actually trying putting a wire underground to see if we can do underground signaling. But maybe we should try using a subwoofer or something along those lines also to signal underneath, because each of these vines, while it's connected via a wire, is also clearly connected through the ground and root system. So scaling up is exactly the next direction. The other thing to consider is each farm has a different Y vine configuration. So I showed you one with a metal post and a T intersection, but you might have a Y intersection. You have heavier and thicker vines that are changing throughout the season. So you're going to have a different water component. So a heavy vine has a thicker stem and stuff that will be cut off come fall. When it's well-watered system, you're going to have a different type of internal dynamic. So all of those are factors. And when you have a lot of foliage, when you have grapes versus no grapes, all of those are factors that are needing to be discovered more before we can say how well it'll scale up. But results, generally speaking, are very promising.
Okay, very good. Thanks, Shira. I think Andrew also had a question for you about your functional analysis plot that you showed.
Mm-hmm.
And so he's asking, is that a form of LVA or ICA? He wasn't sure how you created that plot.
Okay, so what we did is we took the duration of the signal and we basically did an FFT analysis on it to over the whole duration of the signal to get the intensity of each frequency over time combined. So getting rid of the time factor. Does that answer the question?
So I'm referring to the plot where you had grouped together, clustered together, male, female, and sort of the recordings you'd taken and the function that you thought that they were performing.
Ah, right. So how we were able to decide which signals were different from each other?
Yeah. So what was that plot showing? What were the vertical and horizontal axes, and what was
they? Good question. So they are basically, it's a combined factor of different functions from their signaling components. So it could be a mixture of their duration, of their frequency, the dominant frequency of. I mean, we put in many different parameters, and so the computer. I don't recall offhand what the main components of each of those functions are.
So was it principal components analysis that it used,
yes.
or it was just
yes.
it was PCA? Okay.
Yeah.
The take-home message that you can tell.
I think there's a follow-up to that, Shira. I saw in your charts you were showing relative dB.
Yes.
Do you know what the actual intensity of those vibrations are as they go from the shaker to the? Well, I guess.
Yeah, we did some. We're talking roughly to the -6 factor for the vibration factor. So we did this with relative dB, but most of the, we did some using the software itself. And so we're talking at a very, we would hardly feel it. We can feel it with our fingertips, but it's not as much as a millimeter. So let's see, -6 is micrometer level.
Okay, so it's actually low intensity.
Low intensity, absolutely. So we ran these off of basically rechargeable 12-volt battery packs.
What was your question, sir? We have one other question here at UNH. Okay.
I was wondering if I could have the females produce
Okay. Okay, so you're asking, are the females the ones who initiate signaling? Okay. Okay, yeah. So one of Sarah Dodgen here at UNH who was asking about female signaling in the glassy-wing sharpshooters. So they are the ones then that are advertising for mates, and the ones that are the principal respondents to the signal that you're propagating to the grapevine?
It's a duet. So in the beginning, the female usually starts the duetting process and leads that duet. But then after it goes from phase one where they're kind of stationary, then the male is much more in control, and he's the one that's calling more often, investing more energy. What we've seen in the field is sometimes a call fly behavior in leafhoppers, and maybe possibly a call listen, where the male will fly from branch to branch, from vine to vine, looking for female. So he lands, and maybe the female will call, or maybe he will call to identify, is there an animal around, and then he'll fly on. So the female is very stationary in her energetic component, but she does do more of this signaling in the beginning of their courtship. And then the energy switches to the male investing more energy into the signaling, and he leads the second and third part of their courtship.
Okay. So it sounds like what you're saying is that the noise source that you're generating is likely masking the signal?
Not at all, no. So the female is calling, the reason we chose the female signal is partially because she's the one that is leading that beginning phase, but also because the male rivalry signal is very similar to the female call. And so if it were masking, if you remember that one slide I showed where the female is calling next to the female call, so they're actually able to call in response with the playback. So it's not masking their signaling, more of a behavioral influence.
Got it.
And the reason we didn't want to do a straight masking was because there are other beneficial insects that live on the plants, such as lacewings or spiders.
Mm-hmm. Okay, thank you.
I was just curious, I guess both Andy and Shira, they had really nice industry applications. Are they getting any funding from the industry for this, or is it just government funding?
Did you hear the question, Sharon?
Not fully, no. What was it again, please?
I think this was both for you and Andrew. Both of these projects seem to have some promise for industry applications.
Yes.
Getting any funding from industry partners to advance the research? Or is it strictly government funding that's...
Oh, we are funded through a variety of sources. We have some government funding. As I mentioned, the grape industry is very important in California, so we actually have some table grape funding, and some possibly wine grape funding, as well as, our industry partner is working on their end. They try to get funding to work and develop the components. So we're interested in working with them. We focus on the biological side, and as far as the engineering goes, we leave that development up to them.
On our side, we most of the funding for the research I presented was government funding. So there's the through tissue communications was done initially on just sort of internal funds, and then we used that to get some NIH funding for an R21 to do some more animal models. The through soil communications similarly was done internally first, and then we got some funding from the National Science Foundation.
Good. All right, Andrew and Shira, thanks again so much for providing the insights into your projects today. This is, again, our last seminar of the semester, but good news is we are going to reinitiate this in the fall for next year, I think is the plan. So more news coming on that, and I was able to figure out the recording function here for the website soon. Shira, thanks again. Andrew, thanks again. And we will sign off at this point and look forward to seeing you all again in the fall.
Great. Thank you so much for inviting us.
Yep. Thank you. Bye-bye.